Why not? Well, I'm delighted to be here to introduce today's speakers for Yale Cancer Center Grand Rounds. Our first speaker is Dr. Samuel Katz, MD, PhD. He's in the pathology department, and he'll be discussing dissecting and targeting pro-apoptotic BIM in mantle cell lymphoma. Sam, come on up. Okay, you're good.
Yeah, should be good.
All right. So no disclosures. I'd like to thank you for inviting me to discuss my work here. Our lab is very interested in cell death as it's regulated by the BCL2 family of proteins. And today I'll share one story where we're trying to understand its role within mantle cell lymphoma. So mantle cell lymphoma comprises about 5 to 10% of non-Hodgkin B-cell lymphomas. And unlike the other small, mature B-cell lymphomas, is fairly aggressive with a 5-year median survival. There's no current standard chemotherapeutic regimen. And clinically, it involves the GI tract in the form of lymphomatoid polyposis, lymph nodes, bone marrow, and spleen. And molecularly, the hallmark is this translocation between chromosomes 11 and 14. It was actually one of the first B-cell lymphoma translocations for the genes that was cloned out was BCL1, or as we now know it today, cyclin D1. And it led support to this idea that overexpression of a cell-promoting gene like cyclin D1 within specifically these mantle cell B cells can lead towards mantle cell lymphoma. However, surprisingly, when you reduplicate this event within the mice, there's no duplication of the disease. Now, while this was being cloned out around the same time, a second translocation in follicular lymphoma was also being cloned out, and that led to the identification of BCL2. And from that came a large family of BCL2 proteins, which can be broken up into 3 groups. There's multi-domain pro-apoptotic members, like BACs, which are held in check by multi-domain anti-apoptotic proteins like BCL2 through direct interaction and binding. And these interactions are further regulated by a third subclass, the pro-apoptotic BH3-only proteins, which in response to select stress stimuli can bind to a subset of the anti-apoptotic proteins, as in the case with BAD, or can additionally directly bind to and activate the multi-domain pro-apoptotic proteins, as is the case with BIM. And so cancers also take hold of this system in order to survive, perhaps most notably, as we see, as we talked about in follicular lymphoma, through overexpression of anti-apoptotic members, but also increasingly recognized to occur through deletion of pro-apoptotic BH3-only proteins. And as it turns out, both of these mechanisms are taking play by mantle cell lymphoma, where we can see amplification of BCL2 in about 15% of cases, as well as biallelic deletions of BIM in about 20 to 40% of cases. And so this led to a very simple thought that perhaps the cyclin D1 translocation on itself is not enough, and that what we need really is a combination of overexpression of cyclin D1 giving a cell proliferation defect, along with dysfunction of the BCL2 family member giving a cell death defect in order to derive mantle cell lymphoma. And so we set forth to test this within a mouse model, and we first made a cyclin D1 transgenic mouse that expresses cyclin D1 behind the immunoglobulin heavy chain promoter, similar to human mantle cell lymphoma, and got increased expression of cyclin D1 seen by RT-PCR of the spleen, or Western blot of spleen and thymus, or Western blot of splenic B cells isolated and grown ex vivo and stimulated with LPS. But as reported in the past for other transgenic lines, these mice were fairly normal, with normal splenic morphology, fairly small follicles within the spleen that were surrounded by roughly equivalent levels of cyclin D1 positive cells. Now, on the BIM side, rather than using the global BIM knockout, in order to come closer and to approximate human disease, we decided to knock out BIMs selectively within B cells using a CD19 crate. And these mice are very similar to that seen for the global BIM knockout. That is, they have enlarged spleens with enlarged follicles, occasionally have infiltrates of lymphocytes into peripheral organs like the kidney. And these cells are, as you'd expect, B cells that are negative for cyclin D1. So when we crossed these two lines together, we saw a similar lymphocytosis and splenomegaly within both the BIM conditional knockout and the transgenic knockout lines compared to the wild type and the transgene alone, but not much difference between these two. However, when we looked morphologically, we saw only within these mice rare instances of cyclin D1 positive hematopoietic proliferations. And so shown on top is a mouse that has a spleen that's diffusely involved by B cells that are cyclin D1 positive. Next is a mouse that had multiple polyps within its GI tract, similar to the lymphomatoid polyposis one sees in mantle cell lymphoma. Again, B cells positive for cyclin D1. And interestingly, we also found an animal that had this lung nodule that turned out to be CD138 positive plasma cells positive for cyclin D1. And now, while this isn't typical of mantle cell lymphoma, it's worth noting that about 10 to 15% of plasma cell myelomas contain a very similar cyclin D1 IgH translocation as do mantle cell. So, encouraged by at least these rare occurrences, we decided to try to stimulate these mice further to elicit disease. And we decided to stimulate them through their immune system because it's been noted that, in contrast to earlier thoughts, that about up to maybe 3/4 of mantle cell lymphoma exhibit somatic hypermutation and B cell receptor stereotypy, suggesting that there might be an antigen-driven process in the promotion of this disease. And so we used sheep red blood cells, which act as a polyclonal antigenic stimulation, leading to germinal center and plasma cell formation on a cohort of mice with 2 injections, and then checked a little while afterwards. And in these mice, while the spleen weight and mediastinal lymphadenopathy was relatively similar, what was striking was that we saw this expanded population of CD19 positive, CD5 positive mantle cells only within our transgene knockout mice. This didn't correlate with a change in splenic morphology at this time point, so we had both the wild type and transgenic mice having fairly similar small follicles, and the enlarged follicles of the BIM knockout and the transgene knockout mice, again, just kind of lined by a smattering of cyclin D1 positive cells. But we thought that perhaps since we saw this expansion of the CD5 positive B cells, or these mantle cells within this mouse, that if we were to stimulate them a little bit further, we might be able to get disease out. And so after giving sheep red blood cells, we waited till about 7 days when the germinal centers were just starting to form, and hit them with a little bit of pristine. And I should say that when we gave them pristine alone, nothing happened to the mice other than a little bit of lymphadenopathy, but it was non-selective, and it also didn't lead to cyclin D1 proliferations. But in this case, when we gave both of these, we saw in about 1/3 of our mice these large proliferations of B cells that are now positive for CD5, like seen in human mantle cell lymphoma, as well as that do not express CD23 for the most part, which is also commonly seen in mantle cell lymphoma. And now when we looked at the spleens morphologically, we saw sheets of B220 positive B cells that are positive for cyclin D1. And I should note morphologically that these have a very small, mature-looking appearance, their lymphocytes, which is in distinction to all other mantle cell mouse models, which aren't based on the known human genetics, which have more of a blastoid appearance or a large B cell type of appearance. This involved not only the spleen, but also peripheral organs, as shown here, a lung that's infiltrated by these B cells that are cyclin D1 positive. And we went further to try to show that it's not selective just to this treatment regimen, and so we treated with sheep red blood cells followed by irradiation a week later. And again, we saw in about 30% of the mice this expanded CD5 positive population of B cells, negative for CD23, with a small cell morphology that again occupied sheets of the white pulp of the spleen, B cell positive, cyclin D1 positive. Again, it involves peripheral organs, shown here in the lymph node and within the GI tract. And then to go one step further and see if we can get rid of the second cilius and get this just with sheep red blood cells, we decided to treat only with sheep red blood cells, but with a fairly aggressive regimen of weekly doses of sheep red blood cell. And here we saw in about half of our mice now this expansion of the CD5 positive population. In this case, it was positive for CD23, but that does occur within mantle cell lymphoma within humans as well. Again, sheets of B220 positive, cyclin D1 positive lymphocytes that have a small cell morphology. So having defined these two groups, we then went back to try to characterize them a little bit further. And so we looked at this B220 positive, CD5 positive population for clonality and found in this exemplary case that by immunoglobulin heavy chain PCR of the framework 3 region, clonal band for these, but not for the population that's negative for CD5. We also looked at proliferation markers using Ki67 immunohistochemistry and saw enriched, highly increased proliferation within these mice that developed the disease as opposed to the ones that didn't. And this was also confirmed genetically by using microarray, where we took all of the mice that developed the disease and compared it to the mice that didn't develop the disease, and regardless of what treatment regimen they got, and found an enrichment using quantitative SAGE in collaboration with Steve Kleinstein's group for numerous cell cycle profiles, including the reactome cell cycle and reactive cell cycle checkpoints shown here. Looking at downstream targets, such as phosphorylated retinoblastoma, we saw again within the mice that developed the disease an increase in the phosphorylated form of retinoblastoma compared to those that didn't. And this correlates again with the genetic results from the microarray results, where we saw enrichment of genes that are activated or turned on in retinoblastoma knockout, which is what we predict by that overexpression, as well as repression of genes that are turned off when you overexpress E2F. Perhaps most interestingly is that we found enrichment of a signature defined for human mantle cell lymphoma that was derived by Lou Stout's group when they compared mantle cell lymphoma versus other human B-cell lymphomas within our mice, and that if we took that gene set, we could independently separate our groups of mice that developed the disease and didn't using principal component analysis. So this suggests that BIM might play a pathogenic role within disease of mantle cell lymphoma. And not only here, but it turns out that BIM is lost or inactivated by multiple other mechanisms in many other malignancies, including Burkitt lymphoma, renal cell carcinoma, melanoma, CML, or non-small cell lung carcinoma. And it suggests that in order to treat these, we might want to really reactivate this death pathway within these cells. And ideally, we would use the functional portion of the BIM molecule, that BH3 death domain, to hit all of its known targets. That is, the anti-apoptotics that it can inhibit, the reactivation of the pro-apoptotics, and replace the lost BIM. And so to do that, I used a technique pioneered by the Walensky and Verdine labs, whereby they make a stapled peptide. Because if we were to take just that functional domain outside of its native context, it would lose its shape, it's easily degraded, and it's not cell permeable. And so in this technique, we synthesize the peptide amino acid by amino acid, inserting in non-natural amino acids at the I and I+4 positions that contain olefin side chains that undergo ruthenium-mediated catalysis to create a staple. And these stabilized alpha helices of DCL2 domains, or SODs, have marked properties. And since this has been published, I'll just briefly review them. That if you take a native peptide, by looking by circular dichroism, it really has no shape. But if you used a stable peptide, you see this characteristic double hump of a alpha helix. Incubating in vitro with trypsin leads to rapid degradation of the unmodified peptide compared to the stable peptide. And if you put on a FITSI moiety, you can see by multiple mechanisms, but here shown by confocal microscopy, that the stable peptides are now able to penetrate into cells compared to the unstable versions. And so we made a stable peptide corresponding to the BIM BH3 domain, as well as a point mutant that doesn't interact with its known BCL2 targets, and treated a panel of mantle cell lymphoma cell lines and saw a dose-dependent decrease in viability from those cells treated with the BIM-SOB, but not the point mutant control, with EC50s in the low single-digit micromolar range. And this correlated with a concomitant increase in caspase 3-7 activation, again, only by the BIM-SOB and not the point mutant control, in a dose-dependent manner in multiple of these cell lines. So to summarize, I've shown that immune stimulation with sheep red blood cells results in expansion of the CD5 positive B cells only within the transgene BIM knockout mice that may represent a precursor lesion, and that further immune stimulation of these mice can lead to mantle cell lymphoma features as defined by morphologic, immunohistochemical, and molecular features. And that a role, that might suggest, therefore, a role for both antigen-driven proliferation as well as BIM as a tumor suppressor cooperating with cyclin D1 in mantle cell lymphoma. And finally, that BIM-SOB is a pharmacologically optimized peptide that can reactivate apoptosis in cells that lack BIM, and that this highlights the potential role for BIM BH3 replacement therapy in mantle cell lymphoma. I'd like to thank my former PI, where a lot of this work was done, Lauren Walensky, in whose lab a lot of this work was done, as well as lab members that helped out, my current lab, my collaborators, and my support. Thank you.
Useless. No.
Yeah. Okay, well, thanks, Sam. And no one has ever complained about a short talk. So this gives us plenty of time for discussion, though. So I'm wondering if I could start by asking, first of all, what the prospects are for optimizing these peptides into a form that can be used in vivo. And secondly, where they fit in the context of other approaches to pro-apoptotic targeting.
Sure. So first off, I should say that this peptide has been used in a mouse in vivo to treat BIM null proliferations. We didn't have this mouse model set up when we were doing that, and it does lead to apoptosis within that. There is a company, Aileron, that is developing BIM as well as other staple peptides for in vivo use. As you know, there are other BH3 replacement therapies, most notably Abbott's 737 or 263, which targets BCL2, BCL-XL, and BCLW, as well as, I should say AbbVie now, they changed their name, Abbott 199, and that targets selectively just BCL2. The thing that's different about this BIM-SOB is that not only does it target those, but it also targets BFL1, MCL1, on the anti-apoptotic side of things, and it can directly target BAX. Based on that, there are small molecules that have been developed that can target selectively just BAX, as well as people going back now to target the other ones. So whether it winds up going in as a peptide for therapeutics or whether it is used as a tool in which to find and discover more small molecules is yet to be seen.
Okay, great. Thanks, Sam.
I wonder to what extent Thanks. I wonder to what extent your model, I mean, what you've shown us outside of the genetics looks a lot like chronic lymphocytic leukemia, just phenotypically. We didn't see a lot of that. So I'm wondering sort of where in the sort of human low-grade lymphoma thing this most resembles. Genetically, obviously, it's much more like a mantle cell lymphoma. And whether you've tried...
Because chronic lymphocytic leukemia doesn't express cyclin D1, and then molecularly, it shares the signature with mantle cell as opposed to CLL. But you're right that CLL and mantle cell are similar in that those are the two CD5 positive B-cell lymphomas.
And have you looked at any CLL lines or samples in terms of your BIM inactivation?
Peptide towards therapy? Yeah, no, we haven't tried that yet, but one would expect that they could also induce.
Talking about the model as well, was there a lymphocytosis, like is there leukemia that develops in those at all in terms of blood counts?
No, there wasn't as much of a lymphocytosis, and sometimes also in humans it's not.
Well, I'm saying like versus CLL, as opposed to that, it wouldn't, I guess. And I guess from your point of view, looking at the histology as well, it looks like some of this, I mean, may not, it's hard to know if it's polyclonal or there's multiple things, or almost it's proliferative, but not necessarily stochastically taking off to form a really aggressive tumor as much as one might. With the other additions, you got closer. So from a cell biology point of view, do you think what's the chunk that's missing? Because you've added now the anti-apoptotic, you've added cyclin D1. Is there something on the metabolism side or something like that that's not there that's in human mantle cell?
It's possible. There are additional hits within mantle cell lymphoma that occur. Perhaps we're not giving exactly the best antigenic drive for it, but we do see, at least in some cases, that we do get clonality.
So Sam, I don't understand the antigenic boost that you're giving. What do you think is going on in that? You think, and you maintain the presence of the antigen, or is it just a single shot?
So in 2 of the cases, it's a single shot, and in the 3rd case, it's multiple shots.
Right. So how do you think? What's the time course? How long does it take to develop the proliferation? Is it rapid? Is something growing out, or is it, in fact, just a global stimulation of all the cells? And how is that sustained? Is there a genetic change or an epigenetic change that's stable?
Yeah. So possibly a change, but we didn't look for epigenetic or genetic changes. We sustained insofar as that we gave it and we looked 4 months later, so it sustained 4 months past that. But we haven't looked in a large number of mice over time.
So in one of your slides, you mentioned that in non-small cell of the lung, there's inactivating mutation in BIM. Is that a biallelic mutations, or is there loss of heterozygosity or silencing of BIM in lung cancers?
Yeah, I'm trying to remember what that. I don't recall off the top of my head whether it was biallelic or not. I think that from that paper it was actually germline mutations that they found that then led to, in those patients, having resistance.
Any other questions? Okay, well, I want to thank Sam again for a terrific talk.